Hi everyone, this is lecture 16, Cardiac Physiology. So this is the first lecture that is going to start our cardiovascular portion of Unit 3. So we're going to go over and just review the anatomy of the heart. We're then going to talk about electrical activity within the heart and put that together with the mechanical events that happen during the cardiac cycle. As we go through the cardiac cycle, we will talk about some of the equations and some of the calculations that will be done for looking at the volumes of different portions during the cardiac cycle and what the heart is actually putting out in terms of how much blood is being pumped. So that is cardiac output. So we're going to start with an overview of the cardiac system. So the heart is a double-sided pump, and the point of the heart is to establish blood pressure. So as the heart is pumping, it's pushing on the blood, and it's doing that to increase blood pressure and get blood flow out to the tissues. Once it moves out of the heart with blood pressure, then it's going to travel through the blood vessels, which are passageways for the blood to be distributed throughout the body. And the point of this is to get the blood to the tissues where it can exchange materials to and from the cells of the body and deliver the important materials that the body needs and remove the wastes that need to be prevented from building up in the body tissues. We will also do a final lecture on blood, where we will review this liquid connective tissue that transports the dissolved and suspended materials, both the nutrients and other molecules that the body needs, and the wastes that need to be removed. So we're going to start with the heart. So the heart is a large muscle. It forms a double-sided pump. One side of the heart pumps blood to the lungs. One side of the heart pumps blood to the body. The heart is made up of specialized cardiac muscle tissue, but it also contains nervous tissue and protective epithelial and connective tissue layers. So these two sides of the heart make up two circulatory pathways. The first is to and from the lungs. This is a replenishing circuit where blood from the right side of the heart is low on oxygen. It has just come from the body where the oxygen has been used, and is also full of carbon dioxide waste. That blood is then going to travel to the lungs where it can replenish with oxygen and get rid of the carbon dioxide waste. The oxygenated blood then will return to the left side of the heart through the systemic circuit. In the systemic circuit, we're going to get oxygenated blood from the left side of the heart out to the body tissues. So the body tissues need this oxygen for metabolic functions, in other words, cellular respiration. So glucose plus oxygen going to ATP. Metabolism is going to use up the oxygen, and as it does that to produce ATP, it's going to give off carbon dioxide waste. So carbon dioxide waste will be high in the tissues when the tissues are doing a lot of metabolism, and that carbon dioxide needs to be returned to the lungs to be removed. So before it gets to the lungs, though, it has to get to the heart. So the deoxygenated high carbon dioxide blood then from the tissues will return to the right side of the heart, where it will then re-enter the pulmonary circuit and deliver that blood to the lungs. So here's the diagram in your book that shows these two circuits. Here is the right side of the heart, which has low deoxygenated blood, which makes it sort of a more blue-purple color. We'll talk about that later. And that is going to go to the lungs, where it will be replenished with oxygen. When it has a high concentration of oxygen bound within the blood, it turns more of a red color. That blood's going to go to the left side of the heart, where it will be pumped out to the body. That oxygenated blood then will go to the body tissues, where the body tissues can perform metabolism to make ATP. From there, they will produce waste and use up the oxygen. So the blood leaving the body tissues has high carbon dioxide waste, low oxygen, and goes back to the right side of the heart where the cycle will continue. To the lungs to replenish, to the body tissues to be used, back to the heart to be returned to the circuit. So your book also has this diagram where it separates the two pumps as if they were two totally separate structures. So we have then the right side of the heart receiving low oxygen, high carbon dioxide blood through the vena cava, superior and inferior vena cava, and then to the lungs. And then from the lungs, oxygenated blood back to the heart where it can be pumped to the tissues. So I want to draw out the pathway of blood through the heart so we can review a bit of the anatomy and just make sure we're all on the same page with the different chambers of the heart and how the blood is moving through. So let's draw this out. Okay, so the way that this works is we have 4 chambers of the heart. The upper chambers are the atria, and the lower chambers are the ventricles. So starting with the deoxygenated blood, which I'm going to draw here in blue, so this is deoxygenated or low oxygen blood. So it's low oxygen and high carbon dioxide blood is going to enter into the first chamber of the right side of the heart, which is the right atrium. It's going to travel then from the right atrium to the lower chamber of the right side of the heart, which is the right ventricle. From there, the deoxygenated blood will be pumped from the right ventricle out of this large blood vessel, which is called the pulmonary trunk. From the pulmonary trunk, it'll be pumped to the lungs. The lungs are going to take that deoxygenated high carbon dioxide blood, they're going to remove carbon dioxide, and they're going to replenish the blood with oxygen. From there, that replenished blood is going to travel from the lungs to the left side of the heart. And the upper chamber of the left side of the heart is the left atrium. The blood's going to travel from the left atrium to the lower left chamber of the heart, which is the left ventricle. And then the left ventricle is going to pump the blood out of this large vessel, which is the aorta. So from the aorta, the blood is going to be pumped out and go to the body or the tissues. The tissues are then going to use the oxygen, so this blood is going to be oxygenated, which is going to be high oxygen and low carbon dioxide. The tissues are going to use the oxygen for metabolism and produce carbon dioxide waste during the process of metabolism. So the blood leaving the tissues will be this deoxygenated high carbon dioxide blood. That will then return back to the right side of the heart and the cycle will continue. So starting at the right side, we have deoxygenated blood returning to the right atrium through the large blood vessel, which is the superior and inferior vena cava. And then from the right atrium, there is a valve to get into the right ventricle. That's called the tricuspid valve. From the right ventricle, then, it's going to be pumped out of another valve, through another valve, the semilunar valve, into the pulmonary trunk. From there, it'll go to the lungs, and the lungs will oxygenate the blood, returning to the heart via the pulmonary veins to the left atrium. It'll go through the bicuspid valve, which is on the left, into the left ventricle. Then the oxygenated blood will be pumped out of the left ventricle through the aortic semilunar valve into the aorta. So I want you guys to be very, very comfortable with the pathway of blood through the heart. Here's a picture from your textbook, which shows exactly what we just drew. So the deoxygenated blood entering the right side and getting pumped to the lungs, the oxygenated blood entering the left side and getting pumped out to the body. So the heart valves are very important to ensure the one-way flow of blood through the heart. So between the atria and the ventricles, we have what's called the AV, or the atrioventricular valves. On the right side, we have the tricuspid, and on the left side, we have the bicuspid valve. The way to remember this is that tri has an R in it, so the tri is on the right. These are open for blood flow into the ventricles when the atrial pressure gets high. In other words, when the atria get full enough, they will push the AV valves open to allow the ventricles to fill. When the ventricles then pump, they're going to increase pressure in the ventricles, and that's going to close the valves to make sure that blood doesn't backflow into the atria. There are chordae tendineae and papillary muscles attached to the valves, the AV valves, which prevent eversion, or flipping backwards of a valve. The semilunar valves are much smaller and thinner, and they're located in the pulmonary trunk and the aorta. And what they do is they act like little cups. So when the heart is relaxed, or when the atria and ventricles are filling, they're going to cup and prevent backflow during the filling and relaxation phase. So let's look a little closer at the valves here. So between the right atria and the right ventricle, this here is your right AV valve, or your tricuspid valve, and it's called the tricuspid because it does have three little flaps when you look at it from the top-down view. Here's the chordae tendineae that are attached to the valve and then attached to the papillary muscles. So I think of these kind of like an umbrella. So you know how you have strings, excuse me, not strings, but you know how you have wires on an umbrella? So when you flip the umbrella open, then it creates tension on the top of the umbrella to make sure that the umbrella stays in this nice dome shape. If you ever have an umbrella failure when it's really windy, when it flips up, that obviously doesn't do its job, and that would be sort of a failure of the heartstrings as well. So if you didn't have the chordae tendineae, which I call the heartstrings, so if you didn't have the chordae tendineae keeping the valves rooted, then you could have a failure of the valve as the ventricles are pumping. So the ventricle is going to pump and create a lot of pressure. That closes the valve here and makes sure that the blood flows through either the pulmonary trunk or the aorta. The semilunar valves are a little bit smaller, and if you look here at this structure, they look more like a cup. So what they're going to do is they're going to prevent backflow when the pulmonary artery and the aorta are in a low pressure state. So when the ventricles are in a low pressure, when everything is filling, then the blood will cup into the pulmonary semilunar and the aortic semilunar valves and prevent it from going backwards into the ventricles, because we always want blood going towards the lungs or towards the tissues. So here's another picture of the heart valves, just showing that they are a pressure system. So between the atria and the ventricles, when the pressure in the atria gets high enough, when they get full enough, they will push the AV valves open. Then the ventricles will pump to get the blood out, either of the pulmonary trunk or the aorta, and that will close the AV valves, preventing blood from backing up into the atria. So again, there's a close-up view of the valves when they're closed with the chordae tendineae tight on the papillary muscles. and here is a top-down view of the valves. So hopefully you guys did a heart dissection in anatomy, so this isn't too foreign. But if you were to cut open the atria and look inside, down towards the ventricles, this is what you would see. The right AV valve is the tricuspid, which has 1, 2, 3 little cusps. And the left AV valve is the bicuspid, which has 1, 2 cusps. The aortic and the pulmonary semilunar valves are much smaller and thinner and kind of make a little half moon shape, which you can't see from this view, but you can see from the side. So now let's talk a bit about the electrical activity of the heart. I'm going to talk to you about cardiac muscle. I'm going to talk to you about the anatomy of the electrical system of the heart, which is the intrinsic conduction system. And then we're going to talk about two types of action potentials. Action potentials within the intrinsic conduction system that we call pacemaker potentials, and then action potentials in the cardiac muscle, which we're going to call cardiac muscle action potentials. So first, cardiac muscle is striated. Striations come from the arrangement of actin and myosin. If you're forgetting what the arrangement of actin and myosin looks like, you may want to go back to the muscle physiology lecture where we talked about the sarcomere. We do have striations and sarcomeres in the cardiac muscle. The difference, though, between cardiac muscle and skeletal muscle is that the heart needs to function as a unit. So you don't have individual motor units building up in the heart the way you have in skeletal muscle. We need all of the cells in a particular region to be talking to each other and contracting and relaxing as a unit. To do that, in between the muscle cells, we have connections called intercalated discs. They're going to mechanically hold the cells together as the heart contracts and keep it strong so the cells don't pull apart. And then they're going to electrically connect the cells so that any electrical activity in one cardiac muscle cell gets directly transferred to the next door cell so that you will spread activity throughout a whole region like that. There's no synapse here between the cardiac muscle cells. They are directly connected with open gap junctions. So any ion changes in one cell are the exact same as the ion changes in the cell nearby. So here's a picture of the desmosomes, which are these tight junctions, ensuring that as the heart contracts, the cells stay close together. And then here's the gap junctions, which are literally open channels, allowing ions to move and action potentials to directly spread across very quickly across groups of cardiac muscle. Because the regions of the heart are connected electrically by these gap junctions, they form what we call a functional syncytium. So syncytium means that they are a functional unit. So when one cell has an action potential, the action potential spreads to all the nearby connecting cells, and all the cells will contract together. So it'll be zero delay between the action potential in one cell and the action potential in the next door cell. It's going to go all the way through and then everything will contract together. So the atria will form one group or one syncytium, and the ventricles will form another group because we want pumping and filling in the atria, and then pumping and filling in the ventricles. Because of this, it's very important to understand that a heart muscle contraction is all or none. So once you get an electrical signal to get the heart pumping or to get one heartbeat, then that's going to spread through the whole heart, through all the gap junctions in a very specific pattern, all or nothing. The intrinsic conduction system is what causes the electrical activity in the heart and how the electrical activity then will be transferred through the different regions of the heart. So what's interesting about the heart is that it does not require the nervous system to start action potentials. So I'll say that again. It doesn't require the nervous system to start action potentials. Years ago, there was this movie, Indiana Jones and the Temple of Doom. So some of you guys who are a little bit older might know this movie. If not, you should look it up. All the Indiana Jones movies are pretty awesome. But there's this scene in the Temple of Doom where the guy, one of the sort of temple leaders, is sacrificing a guy. And he rips his heart out and he holds it out to the crowd. And when he rips his heart out and he holds it out, even the heart outside of the body is pumping. So that's not totally science fiction. It actually, the heart can pump independent of the nervous system. So action potentials in the heart are generated intrinsically or within the heart itself because of this intrinsic conduction system. It's a set of electrical cells located in different regions of the heart and branching and contacting each other to start and send action potentials through the heart. So the first place is the SA node, and then activity will spread to the AV node. From the AV node, it's going to go down to the bundle of His. From the bundle of His, it'll spread down the bundle branches to the right and left bundle branches, and then up the ventricles through the Purkinje fibers. So let's go through these one by one. We'll start with the SA node. The SA node is located in the upper right chamber of the heart, which is the upper right atrium. This is the fastest of all of the intrinsic conduction system groups of cells. It's going to set the pace of the heart, and its baseline action potentials are 70 to 80 action potentials per minute at rest. Its activity will spread across to both atria and then to the next region of the intrinsic conduction system, the AV node. So upper right atrium, 70 to 80 beats per minute. The next is the AV node. The AV node is located in the lower right atria near the ventricular septum. So this division between the ventricles and between the atria is called the septum. So here's the AV node. It is second fastest, and it's only going to set the pace if the SA node is damaged. So the AV node, if it's acting on its own, if the SA node is damaged for some reason, the AV node can generate action potentials at 40 to 60 beats per minute. Then there will be a pause because of the density of connective tissue where the valves are here between the atria and the ventricles. And then through the septum, you're going to have a little what we call the AV node delay. It's about 100 millisecond delay for the activity to spread from the AV node to the bundle of His. So the bundle of His is down here in the interventricular septum, and it has right and left bundle branches that spread electrical activity down the ventricles, and then from there the electrical activity will spread up the ventricles at approximately 20 to 40 action potentials per minute. So from the bundle to the Purkinje fibers, this is a very slow action potential, but again, it would only set the pace if there was damage to the AV node and damage to the SA node. 20 to 40 beats per minute is too slow to sustain life. It's not life-sustaining. 20 to 40 beats per minute is not enough activity to get enough blood pumping out to the body. So you think of this like a train, and your book uses this example. So the SA node is the fastest, And because of that, the activity of the SA node is going to override the activity of everybody else. So as the SA node is sending out action potentials at 70 beats per minute, it's going to pull all of the others along with it. So in other words, the AV node, the bundle branches, Purkinje fibers, and then the cardiac muscle cells will all be attached to the SA node. So if the SA node is working at 70 beats per minute, then everybody goes 70 beats per minute. If the SA node is damaged or derailed, then the AV node can take over. But the AV node has a speed limit of 40 to 60 beats per minute, so it can't go any faster than that. So it can regulate the heart and cause a heart rate of 40 to 60 beats per minute, or in this case 50. And it will pull everybody else along with it at 50. Now, if the AV node is damaged, then the bundle of His to the Purkinje fibers can set a heart rate of around 20 to 40. Again, not life-sustaining, but they can do it. And then the heart rate in the cardiac muscle cells will be 20 to 40 beats per minute. So all of this is saying that there is a hierarchy to the intrinsic conduction system. The SA node is the primary pacemaker. The AV node can come next. The others can do it, but they are not life-sustaining. So measuring then the rate at which the heart has action potentials and the depolarization and repolarization that is caused by the wave of action potentials through the SA node, to the AV node, through the bundle branches, and up the Purkinje fibers is measured by an electrocardiogram, or an ECG or EKG, initially from the German electrocardiogram with a K. So from here, we have different parts of the EKG that we will be measuring in lab and that you may have seen in other courses. Some of you I know are also EMTs and paramedics, so you may have seen this clinically as well. So the first wave of the EKG is called the P wave, and that is when the SA node sends activity out through to the atria and causes depolarization or excitation of the atria. The QRS complex, or the QRS wave, is when the AV node sends activity down and then up through the ventricles. And this is ventricular depolarization or excitation of the ventricles. The last is the T wave, which is the relaxation of the ventricles or repolarization of the ventricles. So if you watch the activity spreading, the activity starts in the SA node and spreads through the atria. It'll then reach the AV node, where there'll be a slight delay, and then it'll begin to travel down the ventricles and then all the way up the ventricles. This can be traced then looking below at the EKG waveforms. So the beginning of the EKG starts when the SA node fires its action potentials. That depolarizes the atria, and then you'll get your P wave. That will then spread after a little delay up through the ventricles to cause the QRS complex. So this is your P wave. And then this large one is the QRS, the R is the peak complex. And then you'll have another delay. And then this last wave is the T wave, which is caused by repolarization of the ventricles. We're going to go through this in great detail in lab and get you guys some experience looking at these more firsthand. We're going to do this by setting up electrodes, not the 12-lead electrodes that you might do clinically, where you can really get a good view of the heart from many different angles. We're only going to do a 2-lead EKG, but you can put in several electrodes to listen to the electrical activity and watch the waveforms from various different locations in the body. So the electrical activity in the heart can be measured by placing electrodes around the chest and across the arms and legs to capture that activity. So let's look then inside the cells of the intrinsic conduction system and inside the cells of the cardiac muscle and see what is actually happening to set these action potentials. So how is the intrinsic conduction system autorhythmic? So in other words, how does it set the heartbeat? So when we say that the SA node has a pacemaker, that it causes action potentials, how does it actually do that spontaneously without the nervous system? Well, it does that by never stopping and never resting. So the intrinsic conduction cells do not stay at their resting potential the way that a neuron stays at resting potential, right? So a neuron's going to stay at its resting potential and sit there and wait for an input. Intrinsic conduction cells do not sit at rest. So minus 60 would be their resting potential, but they never stay there. As soon as they hit minus 60, their membrane potential starts to drift up and depolarize on its own. This depolarizing on its own is what is unique to the pacemaker cells or the intrinsic conduction system cells. We call this the pacemaker potential. So they depolarize spontaneously on their own until they get to threshold. Once they get to threshold, they will open up voltage-gated channels, depolarize, and then repolarize back down to rest. But again, they won't stay at rest. They will hit their pacemaker potential again back to threshold. So they're continuously cycling. So at minus 60 millivolts, the membrane potential drifts up to minus 40. Their threshold is at minus 40, and that is reached spontaneously. Then their peak depolarization is going to go to positive 10, and then they will repolarize back down to minus 60. I know I haven't told you about the channels yet. I'm doing that on purpose. I want you guys to see the shape of the waveform and memorize these values so that you know what it looks like before you know why. Okay? So here's why. The ion movement during the pacemaker action potentials is unique. So the intrinsic conduction system cells, like the cells within the SA node, have something called funny channels. Funny meaning that they're unusual or strange. So when the researchers first discovered these, they just thought they were weird, so they called them funny. And they're sodium channels. They're abbreviated as IF. So these funny channels are spontaneously open at negative 60 millivolts. So when the cell hits this minus 60 millivolt, these funny channels open. So these are voltage gated, spontaneously opening channels that open at minus 60. When they open at minus 60, they are sodium channels, so sodium is going to enter the cell, just like it would in a neuron. So you have the same sodium-potassium imbalance in cardiac intrinsic conduction system cells that you have in a neuron. So the sodium will enter the cell. When a positive ion enters the cell, it's going to depolarize or cause the activity to increase. From there, you will have transient or T-type calcium channels. So this is a big difference between neurons and intrinsic conduction system cells. The depolarization at minus 50 is due to calcium. And then you're going to have a long-lasting or L-type calcium channels that are going to open at minus 40. This again is another fuller, longer depolarization that's going to get you to your peak depolarization with calcium, not sodium. These will open at threshold of minus 40, calcium will enter the cell. A positive ion entering the cell will continue to depolarize, and you'll get to the peak of the action potential. Then, at positive 10 millivolts, the potassium channels will open, and just like in a neuron, potassium wants to leave the cell. When a positive ion leaves the cell will be repolarized or go back down to rest. But as soon as it hits minus 60 during this repolarization, those funny channels are going to open again, and the cell's going to become depolarized, and it'll reset, and you'll have another action potential. So yes, we need to draw this out, right? Okay, let's draw this out. Okay, so this is the action potential in the intrinsic conduction system cells. So for example, in the SA node cells. So these are the pacemaker cells of the heart. So what's going to happen is you're going to have a negative 60 minimum, at which the cell will never stay. So this is going to be a rest, but the cell never stays at rest. Okay? Once the cell hits minus 60, it's going to begin to depolarize. And this initial depolarization is going to be due to sodium entering the cell. The sodium is going to enter the cell through spontaneous funny channels. That are voltage gated at minus 60. As soon as the cell hits minus 60, sodium is going to enter. That's going to depolarize the cell, and then from there, you're going to have calcium. First, the T-type at minus 50, and then that's going to get you to threshold. So sodium first, then T-type calcium, and then you'll hit threshold. And just like in a neuron, when you hit threshold, you get a huge flood of depolarization. So hitting threshold here at minus 40, you're going to now have more calcium entering, and it's going to be a rush of calcium. You're going to get a big peak, and it's going to peak all the way up to positive 10. Again, these are in millivolts. So this is calcium in through the L-type calcium channels. Calcium is going to rush in, you're going to hit your peak, and then potassium channels are going to open. When potassium channels are going to open, potassium leaves the cell, and the membrane goes back down toward rest. Does it stay at rest? No. No rest for the weary. No rest for the SA node of the intrinsic conduction system cells. As soon as it hits minus 60, it's going to go right back up, and the whole cycle will start over again. Okay, so no rest in the SA node or the intrinsic conduction system cells. Absolutely no rest, and that's because of these spontaneous funny channels that as soon as the cell gets back down to minus 60, it goes back up again. That's like us, right? As soon as you guys have an exam, you have to study again for the next one. So no rest for the weary. Okay, so sodium in first, then calcium to get to threshold, then a lot of calcium for the peak of depolarization, then potassium for the repolarization. From there, from the intrinsic conduction system, we're then going to get activity in the muscle. So just like in the nervous system to skeletal muscle, you have electrical cells causing contraction of the mechanical cells. In this case, you have the electrical cells, which are the intrinsic conduction system cells, causing contraction of the mechanical cells or the cardiac muscle cells. So you're going to have an action potential in the SA node or the other intrinsic conduction system cells, and that's going to cause an action potential and contraction of the cardiac muscle. So now let's look at the action potential in cardiac muscle, and this also has a unique shape and a different pattern of ion flow. So the action potentials in the pacemaker cells, like the SA node, are going to spread to the cardiac muscle cells and cause them to contract, just like a neuron causes a muscle at a neuromuscular junction to contract. But the cardiac muscle cells are also unique. So their resting potential is at -90, and they do rest. They sit at rest and they are waiting for input from the intrinsic conduction system cells. As soon as they get input, they are going to get an immediate and rapid depolarization all the way up to +20, +30 millivolts. Why is it so immediate? Because of the gap junction spread. Very, very, very fast between the intrinsic conduction system and the cardiac muscle cells. So really fast, sodium is going to rush in to the cardiac muscle. You're going to get a huge peak, and then from there, potassium is going to kick in, just like it would for a regular action potential. It's going to start to repolarize. It would go all the way down if the end of the story was potassium. But in the cardiac muscle cells, we also have the addition of some calcium channels. These are calcium channels that kick in at this higher voltage. And they're going to be slow calcium channels that are going to extend the action potential and keep it depolarized for a longer period of time. So potassium starts to go out, and then the calcium says, just kidding, we're not done yet. We're going to make this long depolarization phase, and then potassium can take over and repolarize. So here's the channels. Threshold and depolarization are nearly simultaneous. This is through voltage-gated sodium channels that are going to open very fast and depolarize all the way up to +30. So sodium causes the depolarization phase of the cardiac muscle. Compare that to the depolarization phase of the intrinsic conduction system cells that we just drew. What causes the depolarization of the intrinsic conduction system cells? Calcium. So sodium is back. Sodium is going to cause the big peak depolarization here in the cardiac muscle. And then we'll get a little bit of a repolarization as the potassium channels open. They're going to open briefly at +10 and repolarize slightly. Depolarization is then going to be held or sustained by this addition of calcium channels. These calcium channels are slow and they're going to stay open for a long time. Calcium is going to continue to enter to hold the depolarization in a plateau. Then the potassium channels can take over and cause the final repolarization as potassium leaves. And they will cause the low resting potential as well at -90. So let's draw out the cardiac muscle action potential, and then we can compare the two. Okay, cardiac muscle action potential. Again, we have millivolts over time. They have a low resting potential at -90, and they do sit and wait. So they're going to sit at rest and wait for the input from the intrinsic conduction system. So they're going to sit at rest, but as soon as they get an input, so this is going to be ICS input, so for example, SA node input, then you're going to get an immediate and very fast depolarization. This is going to be due to sodium, voltage-gated sodium channels opening and sodium entering very fast, and they're going to go all the way up to approximately plus 30. From there, you'll get a little bit of a repolarization as potassium channels open and potassium starts to leave. If this was the only story, it would go back down and everything would reset. That's kind of like what happens in a regular action potential, but this is not the only story. At this point now, specialized in cardiac muscle cells, we have some calcium channels. And the calcium channels are going to open to prolong and keep the membrane potential depolarized. So that these slow calcium channels stay open and hold the depolarization instead of it going back down to rest. So we're going to hold depolarization in the muscle cells and then allow the potassium to take over. Then potassium will take over. We'll have potassium out, repolarizing the cells back to rest at minus 90. So note here that there is no after hyperpolarization, that the plateau phase extends the depolarization phase much longer than a normal action potential, and that you have the importance of calcium as well. So compare this to the intrinsic conduction system action potential, which looks very different, right? Intrinsic conduction system action potential starts at minus 60 and slowly rises because of the funny channels. Then its depolarization is not due to sodium, it's due to calcium. And then it repolarizes normal due to potassium. Then everything restarts and starts over again. So I want you to notice as well that calcium has a role in both the intrinsic conduction system and the cardiac muscle cell action potentials. The importance of the calcium in the cardiac muscle is to create this plateau phase. So this plateau phase is important for actually creating a very long refractory period. In other words, the sodium channels inactivate just like they do for normal action potentials in neurons. So they're going to inactivate at the depolarization. That's why you get a little bit of repolarization as potassium kicks back in. But the calcium is then going to hold at the depolarization. This extends the refractory period, allowing a long phase to complete the cardiac muscle contraction. I haven't shown you the cardiac muscle contraction yet, but suffice to say, it needs a long period of time to complete. So you have a very long refractory period in the cardiac muscle cell to allow the completion of cardiac muscle cycle contraction before a new action potential can be regenerated. So the absolute refractory period in cardiac muscle is very long. It's about 250 milliseconds. And the cardiac muscle contraction lasts about 300 milliseconds. So this is not an accident. We need to allow time for the cardiac muscle to contract so that you get a full pumping of the muscle cells before you get relaxation and before you start a new pumping. So the cycle in the heart is pumping, filling, pumping, filling. And you can't get an additional pumping as you're in the middle of this pump-fill cycle, because that would mess up and not allow the full pumping of the blood out. So these alternating cycles of contraction and relaxation in this long refractory period is essential to allow the heart to fully function. So look then at the action potential compared to the contraction. So the action potential holds throughout the time that the mechanical contraction can occur. This is very different from what you'd see in a nerve to muscle, where the neuron would be finished about here, and then contraction would occur. So some cardiac abnormalities that can come from different intrinsic conduction system problems. So bradycardia is slow heart rate. Tachycardia is fast heart rate. Arrhythmias are uncoordinated atria and ventricular contraction. So what we're saying here about this pumping, filling that has to happen. Sometimes you can have damage to the SA node. In that case, the pace will be set by the AV node. That would be a low heart rate at 50 beats per minute. You can also have various types of heart block. So damage to the AV node or to the ventricles, which actually prevents the spread of activity across the intrinsic conduction system. You can also have fibrillation. So this is irregular or chaotic twitching of the myocardium due to possibly some conditions that are preventing this full pumping, filling cycle. So the periods of pumping and filling that we're talking about are periods of contraction of the heart, followed by and alternating with periods of relaxation. So contraction of the heart, this is mechanical now, is systole. Relaxation of the heart is diastole or diastole. During systole, a contracting chamber will eject or pump blood. So contracting is going to be pumping or squeezing. That's going to push the blood out of that chamber. During relaxation, the chambers will relax and fill with blood. So incomplete filling or incomplete pumping or ejection can lead to inadequate pumping of blood to the tissue. So it's essential for the heart to function. So we can then describe all of the mechanical activities during a single heartbeat that are ultimately caused by the electrical activity. So we're going to have the SA node, AV node, etc., which is going to cause the electrical activity and start the heartbeat. But once the cycle starts, everything spreads through gap junctions, and we get a wave of activity through the heart. This wave of activity through the heart dictates what happens during a single heartbeat. So you're going to have electrical activity in the intrinsic conduction system spreading to cause electrical activity in the muscle. Just like a nerve to muscle junction, this is going to be intrinsic conduction system to cardiomyocyte junction, and that's then going to cause contraction of groups of heart muscle. So first we have atrial muscle contraction relaxation, ventricle muscle contraction relaxation, and these have to be coordinated. They will also be coordinated with actions of the valves, and I want you guys to keep track of what's happening in the atria, what's happening in the ventricles, when are the valves open and closed, and what are the different volume levels in the heart. As the heart is filling, volume is going to increase. As the heart is pumping, volume is going to decrease. And then pressure. As the heart is pumping, pressure is high because you're squeezing on the blood. As the heart is relaxing, pressure is low, and that allows the heart to fill. We will also track the heart sounds which occur when the valves close. So we have to start somewhere even though this is a cycle, right? So it's kind of odd, and I think students get very confused with the cardiac cycle. But we have decided, not we, like I was part of the decision, the cardiac cycle by convention starts when the heart is filling. So when the heart is filling, it's relaxing and it's in diastole. So we start everything at mid to late diastole. Then we go through a pumping cycle or ventricular systole. And then we start a new phase of relaxation. So I want to talk to you guys about these terms because I think students also often get confused with ventricular, atrial, diastole, systole, etc. So generally, we have a ventricular focus. So when we say diastole, we mean ventricular diastole. So if I slip up and I don't say ventricular diastole and I just say diastole, or if you see in a textbook mid to late diastole, then that is going to be ventricular diastole. Okay, so diastole or diastole is going to be relaxation. In mid to late diastole, the ventricles are in their mid to late phase of relaxing. So they are going to be filling with blood to their maximum fill. So that is what this picture over here is showing. And both sides of the heart pump and fill at the same time because of the way that the gap junctions are set up. So in this phase, both sides of the heart, both ventricles are completely relaxed. Blood is filling, either from the body tissues or from the lungs. Blood is filling the ventricles. So everything is filling up and getting ready to be sent out. At the end of this phase, we hit our maximum volume that the ventricles can be in. This is called the end diastolic volume, or the EDV. So blood is filling, the atria are relaxed, the ventricles are relaxed, the AV valves are open to allow the blood to enter from the atria to the ventricles, and the semilunar valves are closed to prevent backflow of blood from the aorta and from the pulmonary trunk. So everything's filling. So we start the cardiac cycle with everything filling, and this is mid to late ventricular diastole. At the very, very, very end, to get that last squeeze of volume into the ventricles, the atria contract. So I think of this like when you're drinking a bottle of wine and you sort of get like you're at a dinner party, right? And somebody has the bottle of wine and you get to the point where you just have just a tiny little drop left and somebody always kind of tries to squeeze the bottle to get that last little drop of wine out. That's what's happening when the atria are contracting. So we're very, very close to our maximum fill, and then the atria pump as much from the atria to the ventricles as they can. The average volume at the end of this phase is about 135 milliliters, but we will talk about some variations on these numbers and what happens when end diastolic volume changes. So this is your EDV, or your end diastolic volume. This is the maximum fill of the ventricles. The next phase is when the ventricles receive action potentials to contract. So they've reached their max fill, and now it's time to pump the blood out. At this point, the atria have finished contracting. They've pumped their last bit of blood out to the ventricles, and the atria are relaxed. The ventricles then are going to contract and push. They're going to push against the valves. The AV valves will shut to make sure that the blood doesn't go into the atria. So these are our umbrellas closing. The semilunar valves were still closed from the previous step, and it takes a certain amount of pumping to push open the semilunar valve. So that's why we have two pictures here. So the ventricles receive electrical activity and they start to pump. That forces the AV valves close. That AV valve closing makes the lub of the lub-dub sound when you're listening in a stethoscope. The semilunar valves are initially closed, and with more pressure, then they will open. But they open after the AV valves close. So the semilunar valves are going to open when the pressure is high enough to overcome that. And then blood will start to leave. Blood will be ejected out of the pulmonary trunk and ejected out of the aorta to go out either to the lungs or to go out to the body. So ventricles are contracting and pumping blood. The amount of blood that the ventricles pump out is called the stroke volume. So this is the stroke or the contraction of the ventricles, and how much blood they get out, stroke volume. At the end of this phase, after they get out a certain amount of blood, the heart is not going to be empty. There's going to be some blood left in reserve. So if we could measure that volume, we call that the end systolic volume, or the ESV. The average amount of blood left in the heart after the blood is pumped to the lungs and the body tissues is 65 milliliters. So now we can do some calculations. Are you guys excited? I know, you're excited. So now we can calculate how much blood was ejected in stroke volume. So we know our maximum fill, that was our end diastolic volume. We know our minimum fill, that was our end systolic volume. And the difference between the two, the max minus what was left over, is going to be your stroke volume. So your average end diastolic volume is about 135 milliliters. Your average end systolic volume is about 65 milliliters. How much blood is left? How much blood was ejected? 70 milliliters. So it's just the difference between the two. So max fill, what was left after you pumped, how much you pumped in your stroke volume. And now we can reset. So we pumped the stroke volume out, got the blood out to the body, and now the atria continue to relax and the pressure goes down and the atria start filling. As the atria start filling, the ventricles are also going to be relaxed, and you're going to hit a point when both valves close. The semilunar valves are going to close because the ventricles relax and the pressure goes down in the ventricles. As you do that, blood could backfill, back the pulmonary trunk, back through the aorta into the ventricles, and the semilunar valves are going to cup close to prevent that from happening. As the semilunar valves close, that's when you're going to get the dup or the dub. So the lub sound is the AV valves closing. The dup or the dub sound is going to be the semilunar valves closing. So lub happens during ventricular systole, or when the ventricles are contracting. Dub or dup sound is heard during ventricular diastole, when the semilunar valves close. At this point, both valves, excuse me, both valves are closed. Everything is relaxed. Ventricles are relaxed, atria are relaxed. The atria are filling with blood, but you haven't got enough pressure yet to open the AV valves. When both valves are closed, this is called an isovolumetric relaxation. So you're relaxing, but there's no change in the ventricles. Again, we're focusing on the ventricles here. So isovolumetric, or same volume, refers to the same volume within the ventricles. Once you get enough pressure in the atria, or enough filling of the atria to open the AV valves, we'll be back to the mid to late diastole and back to part 1. So let's go through then the cardiac cycle slowly. Okay, and I think that it would help to probably draw this out. Okay, so we have in the first phase of the cardiac cycle, we're going to have blood entering and the AV valves open. So blood is entering the atria, AV valves are open, blood is filling the ventricles. So the first phase is going to be mid to late diastole. In that phase, you're going to have increasing volume of the ventricles. You're going to have ventricles relaxed, atria relaxed, and the AV valves open. The semilunar valves will be closed. Oops, so I should draw my trunk here, and my semilunar valve closed. And then I want you to note at each phase if anything changes. So the one change that we have in mid to late diastole is that the atria start out relaxed and then they contract. They contract to squeeze more blood into the ventricles, and the volume of the ventricles at the end is going to be EDV, or end diastolic volume. That is your max fill of the ventricle. Okay, so that is the first phase, mid to late diastole. From here, now we're going to get a signal from the pacemaker cells, and that is going to cause contraction. I'm going to show it as like a squeezing. So now the ventricles are going to contract. When the ventricles contract, we're going to call this systole, specifically ventricular systole. So at this point, the AV valves are now going to close. And the pulmonary and semilunar valves will also be closed initially, right? Blood is trying to then get out of the ventricles and pump. So here, your ventricles are contracting, your atria are relaxed, your AV valves are closed, your semilunar valves are closed, the volume of the ventricles, what's happening to the volume of the ventricles? It's going to start out iso, and then it's going to decrease. Why does it start out iso? Because both valves are closed. You have to build up enough pressure for the semilunar valves to go from closed to open. So the volume of the ventricles when both valves are closed is isovolumetric. And then as the semilunar valves, to get enough pressure on them as they open, then the volume is going to decrease. The pressure of the ventricles is going to increase because you're pushing as you're contracting on the ventricles. So at the end of this phase, you're going to have end systolic volume, which is going to be your minimum fill of the ventricles. Then the last phase is going to be everybody goes back to being relaxed. Wow, I just drew a big relaxation. Everybody goes back to being relaxed. The AV valves are still closed from the previous phase. And then blood is filling the atria. So the atria are relaxed, still relaxed. The ventricles are now relaxed, and blood is filling. At the start of this phase, the semilunar valves are closed to prevent backflow because the ventricles now are very low pressure, and the AV valves are closed. They were closed from the prior step. Once we get enough blood filling, the AV valves will then open to allow blood to then fill the ventricles. So we have another phase here where the volume of the ventricles goes from ISO, because you had semilunar and AV valves closed, so ISO volumetric. But once the AV valves open, then the volume is going to increase. So this is a filling phase. From here, as this happens, as the AV valves open and the volume starts to increase, then we go back to number 1 to start the cycle again. So this is step 3, which is early ventricular diastole. Okay, I hope that helps. And there is a diagram in your textbook that shows these different phases as well. So as a review, the intrinsic conduction system of the heart starts the electrical activity. It is the gap junctions then that spread through the heart to cause first the contraction of the atria, which is the blood squeezing into the ventricles and the maximum filling of the ventricles. And then the activity spreading through the ventricles, which causes the contraction of the ventricles and the pushing of blood out of the heart. So we can then combine the electrocardiogram or the electrical activity in the pacemaker cells with the cardiac cycle. So the P wave is when the SA node is active. When the SA node is active, activity spreads through the atria, and that's going to cause contraction of the atria. Then the AV node will be active, and that will have a short delay, and then the activity will spread through the ventricles. As the activity spreads through the ventricles will contract to eject blood, and then the ventricles will repolarize. So the ventricular contraction is the QRS complex that you see here, QRS in red. And then the ventricles will relax to begin refilling and reset, and that repolarization causes the T wave. So you guys have this huge diagram in your textbook. I'm going to link one as well to this presentation, and I want you to go slowly through this diagram, talk it out. Start with the electrical activity. First you have a P wave in the SA node. What happens in the aorta? What happens in the ventricle? What happens in the atria? What happens to volume, and where are the heart sounds? So go one by one through the phases and try to make sense of this graph little by little as to what is happening in the heart mechanically as caused by electrical activity. I have another one here which is a bit more clear. So first you have a P wave. That P wave causes contraction of the atria, or atrial systole. Then you have a QRS wave. That QRS wave causes ventricular systole, or ventricular contraction. Then you have a T wave, which is the ventricular relaxation. Everything resets until you get another P wave. So the atria contract caused by the P wave, the ventricles contract caused by the QRS wave, the ventricles relax caused by the T wave, and then everything resets. You can also follow the volume. So at the end of atrial contraction, you reach your maximum volume of your ventricles, which is your EDV. At the end of ventricular contraction, you've ejected all the blood out, all of your stroke volume out, and you reach your end systolic volume. Then everything starts to refill, and the volume goes back until you hit another cycle. You can also follow the valves and the different phases as we've drawn them. So don't be intimidated by this graph. Just pull it apart one tiny piece at a time, looking at the 3 phases that we've talked about, and try to follow the volume, the pressure, the valve events, the atrial events, the ventricular events, and the electrical events together. The last bit is about heart rate. So the SA node sets the pace of the heart, starting each cardiac cycle as it fires a single action potential. This will begin the depolarization of the atria, and it's going to cause the heart rate at approximately 70 action potentials per minute. Those 70 action potentials will be directly, because of the gap junctions in the heart, directly relayed into 70 beats per minute. The cardiac output then we also want to put together. So cardiac output is going to be how much blood is pumped out of the heart each minute based on heart rate. So we already know how much blood is pumped out in a single heartbeat, right? One single cardiac cycle is going to be the difference between end diastolic volume and end systolic volume, which is our stroke volume, 70 milliliters. Now we know how much blood is pumped out each beat, and we can multiply that by how many beats per minute to get how much blood is pumped out of the heart every minute. So the total output of the heart per minute is how much blood is pumped in a beat and how many beats you have. 70 times 70, 4,900 milliliters per minute pumped out of the heart every minute. The total volume of blood, depending on the person, is between 5 and 5.5 liters, or 5,000 milliliters per minute. So what this means is that the total amount of blood pumped out of the heart each minute is the entire volume of blood through your body. Every single minute, your heart pumps the entire volume, or the equivalent of the entire volume of blood through your body. That's at rest. During exercise, this can increase up to 20 to 25 liters per minute. Why? Because your heart rate and the force of contraction is going to change with exercise. So we can then regulate how much the heart is pumping by changing heart rate, how often the heart is pumping, and by changing volume, or how much the heart pumps. We can alter heart rate through the sympathetic and parasympathetic nervous system, and we can alter stroke volume, or how much the heart is pushing out, through something called venous return. So we're going to take a few minutes to go through each of these, and then I think you guys can be done for the day. So first, the parasympathetic nervous system can decrease heart rate. It's not required, but it can change it, right? So we have the intrinsic conduction system that's going to set the heart rate, but the parasympathetic and sympathetic nervous system can alter it. It's going to do this through the acetylcholine release. Acetylcholine will act through muscarinic acetylcholine receptors to open potassium channels and depress sodium and calcium entry. This makes them more hyperpolarized and slower to reach threshold. That's going to decrease the firing at the SA node, decrease the excitability of the SA node, and decrease the excitability of the AV node, and increase the AV node delay. It will also, if it's reducing calcium entry, it's going to make a shorter plateau phase of the atria, which will cause weaker atrial contraction. So overall, by increasing potassium channel opening and depressing sodium and calcium entry, then we're going to decrease heart rate with acetylcholine through the parasympathetic nervous system. The sympathetic nervous system increases heart rate. We know this, right? During exercise, epinephrine or adrenaline makes your heart pump really fast. It also turns out to pump harder. We'll talk about that in a second. But the sympathetic nervous system releases epinephrine, and this increases sodium and calcium. Increasing sodium and calcium is going to get you faster to threshold, bigger depolarizations, and enhanced opening of these channels. that means that the SA node is going to fire more often. The AV node delay will be decreased, so there'll be less of a delay, and you're going to get an increased speed of atrial and ventricular contraction. So the sympathetic nervous system can increase heart rate. These are some images of what that would look like if you're looking at what kind of action potential is this, guys? Look at it. It has a -60 baseline, but it slowly drifts upward. So are we looking at cardiac muscle, or are we looking at intrinsic conduction system? Intrinsic conduction system, unless you cheated and looked up here. So this would be your normal heart rate set by the SA node, and this would be in red what the action potential looks like with parasympathetic or acetylcholine input. Here in blue overlaid is what the action potential looks like with sympathetic nervous system or epinephrine. So parasympathetic delays and slows the SA node action potential, slowing heart rate. Sympathetic decreases the delay and makes the spacing much faster between the action potentials. So parasympathetic inhibits or decreases heart rate. Sympathetic activates or increases heart rate. So the last bit here is something that we are going to talk more about in the next lecture, but I want to introduce it here while we're already on the subject of end diastolic volume. So what's interesting here is that the amount of blood returning to the heart, or the venous return, is directly related to the force of contraction of the ventricle. So as the ventricle fills from the vena cava and from the pulmonary veins, the ventricles are going to fill, the muscle wall is going to stretch. This filling from the veins going to the atria and then to the ventricles is called the preload. So the amount that the heart fills or loads, the amount of end diastolic volume stretches the wall of the ventricles. And like any other muscle, stretching the muscle tissue creates different length tension relationships between the actin and myosin cross bridges. So in the muscle physiology lecture, we talked about this optimal overlap for the myosin to grab on to the actin. This is the same relationship in the heart, where if it's too stretched out, then the myosin can't grab onto the actin. If it's too close, there's not going to be much of a change. There's an optimal stretch of the heart at which you get a maximum contraction of the heart. So as the ventricle is filling and stretching, excuse me, you'll get an optimal length of the heart wall. Once you reach your optimal length, the resulting contraction will be a bigger maximal contraction. So this is described by something called the Frank-Starling law of the heart, in which increasing the venous return actually causes increased contraction and more blood pumped out, or more stroke volume. This is an intrinsic property of the cardiac muscle due to the overlap of actin and myosin. So this is the relationship here. So the normal resting length of a heart muscle is below the optimal length. So filling the heart more gets it closer to the optimal length. So normal resting end diastolic volume here, about 135. And if we increase that, it gets it closer to optimal length. We're going to get a bigger contraction. So we increase end diastolic volume, we're going to get bigger stroke volume. This is just called the Frank-Starling curve, and this describes the relationship. And actually, you can get this continued pursuit towards optimal length through huge numbers of end diastolic volume. And the cardiac muscle doesn't actually normally go beyond that. It's very rare that we would get past this sort of 300 milliliter range for optimal length. So the last thing is that the sympathetic nervous system can not only increase heart rate, it can also increase contractility of the heart. So it can increase stroke volume as well by making the heart pump stronger. It does that by getting more calcium to the muscle. When you get more calcium to the muscle, just like in skeletal muscle, you can increase the number of cross bridges formed within the cardiac muscle. You don't get tetany in cardiac muscle because of the limits on the refractory period, but you can get more cross bridges and overall a stronger contraction force. So we can increase stroke volume by increasing venous return and by increasing sympathetic nervous system activity. So here's the role of the sympathetic nervous system, which will increase heart rate, increasing venous return, so more blood filling the heart, increases the end diastolic volume. It can also directly increase contraction by increasing calcium inside the cardiac muscle. Both of those are going to increase the stroke volume. I know this sounds like a lot right now. We're going to do more of this in the next lecture. So this last piece is where I'm going to leave you guys today. You have the normal end diastolic volume of the heart. You would have the average end systolic volume of the heart, which would give you a stroke volume of about 70 milliliters. If we then need to increase the stroke volume, we can add sympathetic stimulation. Just adding sympathetic stimulation will increase the contractility of the heart through increase in calcium to the heart muscle. That will increase the stroke volume to say from 70 to about 100. When you do that, you can also compound on it to add the Frank-Starling law. So not only increasing the sympathetic stimulation, but also increasing the end diastolic volume. When you increase the end diastolic volume by pumping more and filling more in the heart, then the Frank-Starling law kicks in. We get closer to optimal length, and we compound even bigger increase in stroke volume. So sympathetic nervous system increase on contractility does some. Sympathetic nervous system plus optimal stretch of the heart through venous return, even more increase in stroke volume. All right, enough for today. We're going to put this together for you guys in the next lecture where we will talk about cardiac output again. But remember, for cardiac output, we can affect the heart rate through the nervous system, sympathetic and parasympathetic activity, or we can affect stroke volume directly through sympathetic activity or through the optimal stretch of the heart through venous return and end diastolic volume. All right, take a really good break, you guys. Come back and we will do the blood pressure lecture.